Hey guys, Rouge here. So if you haven't noticed, I am out today, but I'm making this little video so you guys get to know what's going on. You don't fall behind with the other block periods. But essentially, what I'm going to have you guys do is I'm going to have you continue to work on your Meiosis iMovie. This is the first part, which hopefully should be done already. Okay. If you haven't finished the Meiosis part, please knock that out before you start on Meiosis. But now what you guys are going to start on, after you're done with the mitosis portion, you're going to start on the meiosis portion, okay? So let me refresh your memory. Mitosis is forming new body cells. Meiosis is forming new sex cells, okay? Or new gametes. So you're going to need all the same materials as before, except now you're going to need little beads that are 2 colors of every chromosome, okay? So you're going to need 2 colors of every bead. And you're going to use these beads to show crossing over, okay? Or how, variation is formed in the gametes. So same materials as before. You're going to need 2 beads of every color. You're going to show a title page, okay? So just give me you and your partner's name again. And now let me know, so as I'm grading this, put in there meiosis, okay? So now that I know that you're done with the mitosis part, and now you're moving into meiosis, okay? That's going to be your title page. Again, you're going to show interphase, okay? You're going to show interphase, and actually for this part, if you just want to copy and paste this portion from your mitosis video and paste that in, that'll totally work because it's the same exact process. Interphase works the exact same way in meiosis, okay? After you're done with that, this is where it starts to get a little bit different. You're going to start meiosis 1 because remember, gametes go through 2 cell divisions, and they go through 2 cell divisions to end up with 4 different cells that each has 23 chromosomes, okay? So it's going to go through the whole process twice, and you're going to end up with 4 cells. There you go, so you can see my fingers. 4 cells. So first, prophase 1. This is the most important part of meiosis, okay? Prophase 1. What you're going to do is in your nucleus, you're going to show the chromosomes, okay? You're going to show the chromosomes form a tetrad, okay? So this structure, when the 2 homologous chromosomes come together and intertwine, that forms the tetrad, okay? Now, I don't know if you can see on here, I have the red bead on the red chromosome, the blue bead on the blue chromosome. What those represent are different genes, okay? So take a snapshot of every homologous pair as a tetrad, okay? So your green and white, they're also going to form a tetrad as well, okay? What you're going to do, still in prophase 1, you're going to swap the beads, and this is going to show genetic recombination, okay? Or new combinations of traits. Just like that, okay? So you're going to swap the beads to simulate crossing over. Now I have a new combination of traits on each chromosome, okay? And you're going to do that to both tetrads, okay? So you're going to show the tetrad and then show crossing over. And now this chromosome, every time I show this chromosome in a picture, it's going to have that green bead on there. It's going to have that variation, that new trait, okay? Same thing with this one. Every time I show this chromosome or this chromatid, it's going to have that variation, okay, or these genes right here, okay. When you move into metaphase 1, metaphase 1 is pretty much the same as metaphase and mitosis, except now instead of having your chromosomes line up in a single file line, remember the chromosomes line up as the tetrad still, okay, they line up side by side. You're going to go to Anaphase, okay? Instead of pulling apart individual chromosomes, it's going to move the entire chromosome, okay? The duplicated copy, okay? Go ahead and show telophase 1, cytokinesis 1, and then you're going to start meiosis 2, okay? So let me clarify one part. When you start prophase 1, prophase 1 should start with 1 cell. This 1 cell is going to split into 2 at the end of cytokinesis 1, okay? So at the end of cytokinesis 1, you should have 2 cells. Those 2 cells, just like on your little storyboard that we worked on, those 2 cells are going to go through meiosis 2 together. So when you start prophase 2, you should have 2 cells on your whiteboard. And then when you go to metaphase 2 or metaphase 2, you should show 2 cells going through metaphase 2. What I'm looking for is the end result that all 4 of those cells are different. They all have a different combination of genes. Okay, they all have different chromosomes in their nucleus. Refer back to the other link that I posted that covers the lecture from Tuesday, and that will help you up with the animation. Okay, because I put the animation on there. I have to have these explained, these questions. Okay. At the end of meiosis, I have to have these questions explained, and also I have to have these questions explained at the end of mitosis. Okay. So follow this outline to a T, and that is your pathway to get an A on this assignment. What I'm going to have you do at the very end, we'll do it in class. I'll show you guys how to do it, but I'm going to have you upload it to YouTube, and then you guys could just submit the link to me, and that'll be the easiest way for you to turn it in and for me to grade it. Okay. If you guys have any questions, shoot me an email, or I'll see you tomorrow. Okay. And please behave, be respectful, clean up your mess. All of your materials, all the beads, just go ahead and give it to the most responsible person in your pair, and you guys take them home. And if you want to, you could work on it on your own time. Okay. And I will see you guys on Friday. Adios.